What does a gene, such as an opioid receptor, do? What is its function? To try to answer this question scientifically, one can approach this through different means. One being, where is the gene expressed? Where is the protein produced? Some proteins are produced in every cell, and some in only specific parts of the body. So if one were to look for expression of opioid receptors in the human brain by using radioactive antibodies which bind to the receptors and therefore would allow you to visualize the presence given the radioactive antibody which bound to them, one could then ask the question, where in the human brain are opioid receptors expressed? If this simulation represented your data from the antibody binding, what would you presume about the expression of mu opioid receptors in the human brain? Are they expressed everywhere? Only in specific regions? If one knew the function of brain regions where the expression was highest, for example, pain processing and reward pathway processing, how would one interpret this data? Often it is easier to study something in an experimental animal, such as a mouse, than in humans. If one performed the same experiment in mouse brains, what would one conclude from the expression of mu opioid receptors given here? If one could identify the function of the brain regions where the expression is highest, and once again, perhaps link them to pain processing or reward processing, what might you conclude? There are different opioid receptors. The mu opioid receptors represented here in red, the kappa in yellow, and the delta in pink. If one observes this expression pattern, what conclusions would one draw about opioid receptors in the brain? If the expression patterns in humans and mice are similar in some ways and different in others, what conclusions would you draw about opioid receptors and mammals?